All right, so today I'm going to show you how to write a kick-butt conclusion. You're going to need the skeletal outline for how to write a conclusion. You're going to need a student example for how to write a conclusion. And you're going to need at least 2 different color highlighters. If you have more, that's awesome, but if you only have 2, that's fine. Okay, let's get going. So, when we talk about conclusion writing, Mr. Owen says conclusions are the most valuable part of a research paper, and I have to agree with him. This is where all the hard work that you've put into designing a really great experiment and then doing it and collecting all of the data and analyzing it, it's the conclusion where you finally come up with all of the meaning. And this is like the reward for all of your hard work. So we're going to look at a conclusion that was written by one of my students, and it's titled, "What are the effects of salt concentration on dandelion germination?" Now, why would he be looking at that? Well, notice at the top that nice, beautiful yellow flower. Dandelions aren't so bad looking. As a matter of fact, they're kind of pretty. And then when they go by, take a look at the beautiful, fluffy flowers, that big sort of otherworldly little ball of fluff. Every single fluff is attached to a seed. So one dandelion can create hundreds of new dandelion babies. Well, if you are Mr. Martin, that means that you're waging war with dandelions on your lawn. He has a gorgeous lawn and he hates dandelions. So we were talking one day about what he could do to control the dandelions growing in his yard. So I spoke with my students about taking dandelion seeds that I had collected, and they wanted to see if watering those seeds with different concentrations of salt would impact their ability to germinate or grow. We're going to take a look at a really nicely written conclusion that was crafted by one of my students. And you're going to notice that that conclusion has 6 key parts. You're going to find this in your skeletal outline. The first part is to restate the purpose of the experiment. Second, analyze the data. 3, compare your findings to the hypothesis. Your findings are the data. 4, discuss possible reasons for error if you suspect that error. 5, final conclusion. What can you really say about that experiment? And 6, if you were going to take this experiment one step further, what might those future studies be? What would you do? What direction would you take your investigation? Now, we've made a mnemonic device to help you remember these 6 key parts, and it's race for food. It may not make a whole lot of sense to you, but maybe you could imagine a good experiment with rats, and they're racing to get to the end of the maze for food. But this is the end of the experiment, writing a conclusion. So let's get right to it and look at the student conclusion. The first step, restate the purpose of the experiment. Okay, the purpose of the experiment was to determine the effect of salt concentration on dandelion seed germination. Now, click pause and take a moment to highlight only this section of your sample student conclusion. In the margin, use your highlighter to write R for restate, because you're restating the conclusion. Next part is analyze the data. Again, you can complete the skeletal outline portion of your notes, because when you analyze the data, you are looking for trends or patterns or relationships. What happens to the slope of the line in the graph? What happens to the data? Does it get 2 times as large or half as large? Be specific. Include certain actual data, but there's no need to repeat all the data in words. You only talk about trends or outstanding pieces of data. Talk about your thesis statement that addresses the IV and the DV. In this case, remember the IV is the salt concentration and the DV is the dandelion seed germination. Support your thesis or your statement with specific examples. So, let's take a look at the next section in the student conclusion. Now, all of this is the data analysis section. It was concluded that as the salt concentration increased, the germination decreased. A 1% salt solution seemed to reflect minimal impact. However, a 2% salt solution caused a considerable reduction in seed germination. The change was even more drastic when the concentration was reduced to 4%, where the number of seeds germinating was nearly 1/4 of the seeds that germinated in pure water. This is shown in tables 1.2 and graph 1.2. They show that the salt concentration did indeed have a negative effect on the germination of dandelion seeds. So each section of this data analysis here is in a different color. However, what I'd like you to do is click pause and take a moment to highlight using a different color highlighter this section of your sample student conclusion. In the margin of your highlight, use your highlighter to write A for analyze, and you're analyzing the data because this whole section is data analysis. Part 3, compare the findings to the hypothesis. This is where we take a look at our data and compare it to our hypothesis. Was it supported or not? And this is where we talk about why it's important. So if we go to our student example, it says the hypothesis stated that as the salt concentration increased, then the dandelion seed germination would decrease. This hypothesis is supported by the gathered data. When the salt concentration went from 0 to 7%, the average dandelion seed germination decreased from 4.67 seeds down to 0. This is likely due to the cells within the seeds becoming more hypotonic as the salt solution increased. The cells were likely dehydrated and died. It's also possible that salt diffused into the cell and interfered with cellular homeostasis. Wow, this is awesome. Not only did this student talk about the data and what happened as a result of an increase in salt concentration, but the student went back to remembering everything that he learned about osmosis and diffusion and talked about how that's applicable in this particular experiment. That's kind of cool. Now, click pause and take a moment to highlight. You're going to change your highlighter color. Highlight this section of your sample student conclusion. In the margin, use the highlighter to write C for compare, because this is where you're comparing the data to the hypothesis. Fourth part, possible reasons for error. This is where, when we're concerned about our data not seeming right, or we're just taking a look at what could have gone wrong that might lead us to question the outcome of the experiment. Let's take a look at what our sample student conclusion says. There are many areas of this lab where error could have occurred that would have changed the results, though there do not appear to be any obvious error in carrying out the procedure properly, and the data is supported by solid science. If the same dropper were used for every solution, this would have affected the error because it would have changed the concentration of the salt solution. Another thing that could have happened would be confusing the salt concentrations put on the seed groupings. However, the lab done by one group in the class had one issue. The seeds did not germinate. This could have been caused by either not following the procedure correctly or by having a bad batch of seeds. The teacher did give that group the data collected by last year's biology students to use. This, in fact, is the case. We had one group whose seeds did not germinate, and I did give the students a previous year's experiments data to use so that they had data to work with. Now, I'm really not quite sure what happened. I'm pretty sure it's that the students did not follow the directions properly because everybody in that class used the same group of seeds. So for that one group to have something goofy happen, but we needed some data to work with, so that's how I chose to handle it. You may be comfortable with that or not, but that's what we did in that particular day. Now, click pause and take a moment to change your color of highlighter and highlight this section of your sample student conclusion. And in the margin, use your highlighter to write E for error analysis. Now, before we go on to the next section, I want to talk about possible reasons for error and what would have happened if there really was something that didn't look right. What if there clearly was something wrong with either how the experiment was conducted or there was something really unusual about the data that was collected? Now, this is not in your sample's conclusion, but just think about this for a minute. The data collected from this lab does not make sense. This makes one question the validity of the experimental design, how the experiment was conducted, or the science considered when forming the hypothesis. All three of those are possible if data was collected that just flat out didn't make sense. So, a student might write, less than 75% of the seeds germinated. At this point, it cannot be determined if this is due to poor growing conditions, which could be overwatering, poor soil, inadequate light or heat, or possibly outdated seeds. Unfortunately, due to a lack of meaningful data, a reasonable conclusion is able to be made. That is a perfectly acceptable concluding statement. A reasonable conclusion is not able to be reached. That's absolutely fine. Part 5, you're finally making your last statement about what you conclude about the experiment. Now, in conclusion, seed germination decreases as the salt concentration of the water they receive increases. Table and graph 1.2 both support this and show us the drastic decrease in the germination of the seeds as the salt concentration increased. Notice that you don't have the word us in there, but I kept it in the slideshow and I put it in red. Why do you think that that is in red? Right, you're not supposed to use personal pronouns. Good job. So click pause and take a moment to highlight, again, change your color, highlight this section of the student conclusion, and in the margin use your highlighter to write F1 for final conclusion. Further studies, last part. This is where you take a look at what would happen if you were to take this experiment one step further. What would you change? What would you change about the IV or what would you change about the DV? In this case, our student wrote, an area for further study would be to investigate how salt concentration affects other species of plants or to investigate the effect of other solutions on the dandelion seed germination. So click pause and take a moment to highlight, again changing color, this section of your sample student conclusion. And in the margin, use your highlighter to write F2. This is your further studies. Finally, last notes. Make certain when you're writing a conclusion that it's in third person. Never use personal pronouns. So you don't use I, we, they, and the conclusion is written in the present tense. That's it. If you follow steps 1 through 6, you're going to write an awesome conclusion.